Scientists remote-controlled designer human cells implanted in mice using only DC currents. This is by Dr. Alexandrana Daguspa. Using off-the-shelf consumer batteries, scientists stimulated insulin release from engineered human cells implanted in diabetic mice and restored the rodents' to normal blood sugar levels. The gene activity of designer human cells can be remotely toggled on and off with electric currents, the new study in mice shows. The study, published July 31st in the journal Nature Metabolism, direct current, or DC, from off-the-shelf consumer batteries triggered the release of insulin from genetically engineered human cells that scientists had implanted under the skin of diabetic mice. The insulin successfully restored the rodents' normal blood sugar levels. The researchers hoped this electrical fine-tuning of gene expression, broadly known as electrogenetic technology, will eventually be integral into wearable devices that can be used to tune the activity of designer cells implanted in the human body. Wearable devices are already in vogue and can monitor pulse, blood pressure, blood sugar levels, and more, but currently no such tech can be used to control gene expression. To move this idea from science fiction to the real world, Martin Fussenegger, a professor of biotechnology, bioengineering at Zurich and the University of Basel, and his colleagues designed an interface called DC Actuated Regulation Technology, or DART for short. It's powered by DC from standard 1.5 volt AA or AAA batteries. As a proof of concept, they tested DART in a mouse model of type 1 diabetes. They implanted engineered human cells in the rodents' backs and stimulated the cells through 2 acupuncture needles placed near the implantation site. The needles were attached to the batteries via wire, and whose end was plugged into simple power switch. The electrical current flowed through the needles and triggered minor oxidative stress in the designer cells, meaning it caused a slight buildup of reactive molecules called reactive oxygen species, or ROS for short. These molecules were detected by a molecular sensor built into the cells. The sensor, which was engineered to work as a transcriptor factor, a protein that latches onto DNA to turn a gene on or off, then bound to the designated spot on the cell's DNA and in turn activated the gene of interest, the insulin gene. The human cells were genetically engineered to express or activate the gene of interest only at the ROS level, produced by the electrical current were high enough, and as the ROS dissipated, the gene switched off. Stimulating the designer cells for just 10 seconds once a day was enough to induce gene expression and trigger sufficient insulin release to restore normal blood glucose levels in the lab mice. Fussenegger told Live Science he thinks this new electrogenetic interface is a complete game changer. He says it's a tremendous application of electrogenetics, a technique that uses electronic means to turn on specific gene expression. William Bentley, a synthetic biologist and professor at University of Maryland, who was not involved in the research, told Live Science the Fussenegger lab previously designed an electrogenetic device that used alternating current AC at high voltage to activate cells, but it required too much power to be sustainable for wearables. And this is on Live Science by Dr. Alaknanda Dasgupta. Please leave your comments. Thank you for your support. My Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.